We have studied about the metabolites. Then now it's a time to combine them together as a big cluster that can be metabolomics. Okay, so far we have studied the genomics. In human case, maybe above 20,000 of genes. And transcriptomics is a RNA sets which can be transcribed from genome. It can be more than 100,000 of transcriptomics for the human, right? And proteomics, it can be different. The translational, post-translational modification can make huge diversity of protein sets. It can be maybe more than a million. Now we are getting into the another OMS, metabolome. So each individual metabolites all together, if you monitor all the metabolites together, those sets we call metabolome. It's equivalent to the genome, transcriptome, proteome. Now we call metabolome. Well, one good thing for you, maybe, the metabolome number is not like millions. So usually, depending on those counting methods, maybe 2,000 or up to 8,000, that's kind of the guessing of those metabolome numbers. So those metabolomes are small molecule generated by our body, and then all together those molecules as collections. If we can monitor them, then a lot of different things is possible. Now here's one small the review paper and also the example of the metabolome measurement. In this case here, this example shows that this is the animal-based metabolome. I guess you may have some experience of taking animal spectra for one or two compounds. But now here we are talking about by collecting all those metabolites from one sample, either cell or tissue, and then take those mixtures of animal. It can be quite messy like this, right? So before we continue, let's hear some experts' opinion about this, and then continue.
By 2020, half of Americans will become diabetic or pre-diabetic. It's a staggering number. Diabetes is due to the fact you have aberrant metabolism, and your collection of metabolites, if you will, is in fact the metabolome. Metabolites are these key molecules, are the chemicals in your body that let you do all the things you do. It's the glucose, it's the ATP, it's all these things that give you energy, that let you move, that let you think, that let you digest your food. So much like a car uses gas to power itself and move, we have chemicals that do that, and it also has chemical byproducts that actually get spewed off that are the waste. Now the amazing thing is we can follow all these chemicals and try and get some understanding of people's health state. The mass spectrometer is the instrument they use when they analyze a drop of blood for certain metabolites to make sure people's metabolism is fine, but we also analyze your urine where you can look at secreted metabolites. So controlling your metabolite levels is very, very important. For me it's glucose, for other people it's cholesterol, and for others it may be things we don't even know about yet. So we are trying to discover the whole collection of these molecules. As we follow Scott and Mark through their journey, both in space and down on Earth, we hope to see this whole collection of changes that occur as people undergo space travel. We know that some chemicals are basically indicative of high stress, and others, we don't know what they do yet. But by following Scott, we hope to see which stress molecules are getting activated and at which times, and what other kinds of metabolites might be happening as he travels on his journey through space, and how does it revert as he comes back to Earth. Mark will be down on Earth and he'll be the control for this. We'll be able to compare the two and see how their changes differ and are the same. One interesting aspect of this whole study is that Scott is actually under a very defined diet. And your diet definitely controls your metabolome and all your metabolites. So we'll be able to follow which foods cause what kinds of metabolite changes and how that changes over time. it'll be very informative to see exactly how what you eat determines what you are chemically. And so it's going to be very, very interesting to see how this all plays out. At the end of the day, metabolomics is a really important field because it actually lets us follow molecules that are very directly important for our health, and it also is something we can control through our diet and other means.
Okay, welcome back. So I hope you got some big the picture of those omics from genomics to metabolomics. Now metabolomics we can say this is a systematic study of a very unique chemical fingerprints. That's kind of a keyword here. Then using this information, this is another kind of the big data, and then we can get complete view or much more comprehensive view. Now this metabolomics is based on from the information of protein. So those genomics information make out eventually proteomes. And then these proteomes, especially those enzymes which is involved in metabolism, and they generate a lot of these primary metabolites such as amino acids and carbohydrates or lipid molecules. And also secondary metabolites which can be used as a natural product and drug candidate molecules which can affect different species. So using this the metabolomic approach, we can get omic the view, then what kind of changes is happening in the whole cell level or even whole body levels. In previous the lecture, we talked about utilizing NCI60 cancer cell line as profiling of compound effect, right? And also transcriptome analysis, utilizing those mRNA change information, then also we can utilize that kind of the monitoring. Now, metabolite, utilizing those metabolite changes, it can be direct metabolite from the introduced drug molecules. Drug molecule can get into the cell, get into the body, and then it can be metabolized and generate different the direct metabolites. It can be maybe, you can say, primary metabolites in this case, for that the drug at the viewable point. And then it's a secondary effect of changes of metabolites in the body that can be used as a profiling, another profile which can tell the mechanism of drug molecules. Then let's see what is currently possible in metabolomics. To summarize again, metabolomics is chemical fingerprinting. We can take out the old chemicals from the biological system and utilize those fingerprinting of changes of those metabolites all together. Use that information to understand the biological the events. Several different techniques, but most likely, let's say, if you utilize NMR, one of the limitations is the sensitivity. Using high level of the magnitude, the magnetic the field of the NMR, still we need micromolar concentration of those material, sometimes millimolar concentration. And one molecule can generate multiple peaks. That's why practically maybe approximately 50 metabolites we can monitor practically at a time. Gcms is another very popular technology for this. And sensitivity can be slightly better than NMR, but still the detection the detectors the mode still kind of a sub-micromolar concentration we need. And not so many metabolites can fly in the gas chromatographic condition. That's why approximately 70 to 100 metabolites can be detected by the GCMS. Many of those water-soluble metabolites, HMS may be more popular method, and also sensitivity can be much higher too. Some of those concentration effect and also utilizing high sensitivity of mass spec. Now using this HMS, maybe you can detect several hundreds of metabolites together. So this is currently possible the mode of metabolomic studies. In addition to the experimental the setup, also another important features should be the informatics side. So here I introduce you for the first time one the good database for the human metabolome the database. So to encourage your usage and experience of this, so here's your second the quiz material. You can choose your own interesting one metabolite and try to get out as much as the useful information from this database, and then send me that result as your the quiz answer to the poll in Ivy League. There's a different view of this the database, so that's why I also seek one technical talk volunteer to introduce how to use, efficiently use, and what can be the important the result using this database. I want to invite one the tech talk volunteer, so you can go to the poll. Again, I will put the collection of this the tech talk volunteer. Then this is also another option, and the earliest the volunteer will have a chance to present in the next class. That's the next Monday class. Okay. Now this is the last page of this week's lecture, and again you have a chance to ask any question from this lecture and any unclear part in the lecture, and you can give me the give me your other question, and also any other extended topic as a candidate for the tech talk. Then we can discuss this in the class together. So from last week we changed the deadline. You remember that, right? So deadline of getting submitting all of these the questions, the answers for these questions are the Sunday noon before the class on Monday. Okay, so enjoy everybody and see you in the next class. Bye bye.